So now sex determination. Determination of a sex at the time of fertilization. So because in the fertilization, two different gametes fuse to form a zygote, so there might be a change in the sex. So the determination of sex at the time of a fertilization is known as the sex determination. The chromosomal pattern in a human female and human male is different. Like in a human female, two X chromosomes are found in a sex chromosome. So as a sex chromosome, two X chromosomes are found, and in a male, X and Y chromosomes are found. So therefore all the haploid gamete produced by the female have only sex chromosome, that is X, whereas in the male gamete, the sperms, the sex chromosome could be either X or could be either Y. Now next, hence in the male 50% of the sperms carry the X chromosomes while the other 50% carry the Y chromosome, while female has 100% X chromosomes in its every gamete. After fusion of the male and female gamete, the zygote would carry either XX or XY depending on whether the sperm carrying X or Y fertilized with the ovum. The zygote carrying the XX would be developed into a female baby and XY would be a male. It means, what it means to say that after fusion, the male and female gamete fuse to form a zygote, and the zygote would carry either XX or either XY. It will carry either XX or either XY. This is XX or XY. Depending on whether the sperm carrying X or Y fertilized to the ovum. It means the determination is based on the sperm carrying which type of a gamete. It means the determination is based on the male gamete. It means the determination is based on the father. The sex determination is based on the gamete produced by the father. The zygote carrying XX would be developed into a female baby and XY would be a male baby. That is why scientifically it is correct to say that the sex determination of a baby is determined by the father and not by the mother. The similar thing it was saying. Now we can see from here, the male is having a two set of a sex chromosome, one is X and one is Y, while the female is also having a two set of a sex chromosome and both are X chromosome. So when this male is forming its gamete, one of its gamete will be having X chromosome and one of its gamete will be having a Y chromosome. While when the female is producing gamete, both of its gamete would be having either this X or either this X. Means the gamete of female can only have X chromosome with it. So a female will also form two gamete. Means if you can form it here, this gamete is also containing X. One X from this and one X goes to this. So female also forming a gamete, but both the gametes are having only X chromosome within it. It means the male can form two different types of a gamete, one with the X chromosome and one with the Y chromosome, while the female can only form only one type of a gamete. Both the gametes are of X chromosome. So the female can be producing only one type of a gamete while the male is producing two types of a gamete. Now, when this gamete of a male fuse with this gamete of a female, the offspring will be having one X from the male and one X from the female. Means one from the mother and one from the father. And this offspring will be a female because it is having two X chromosome. Now the next, if this X chromosome of a male if this gamete of a male fuses with this particular gamete of a female, it will again be forming 2X. So it will again be a female child. So whether this gamete or whether this gamete, any of the gamete female fuses with the X gamete of a male, it will be forming only a female child. So any of the gamete means the female gametes are not been providing any change in the determination. Now if the Y chromosome of a male fuses with this X chromosome, it will form XY, while if this Y fuses with this X chromosome, it will again form XY. So this XY will be a male. So why this would be a male? Is any of the female gamete has produced any kind of a change in this? Female can only contribute the X chromosome, while the male this Y chromosome comes from the male chromosome, and this Y, because of this Y chromosome, this particular baby, this offspring will be becoming a male. So only the Y chromosome is responsible for forming a male. This X chromosome, the female is contributing only one kind of a chromosome, but because of the Y which came from the male gamete, this body, this particular organism will be a male. So sex determination should be based on only the male gamete. It should not be based on the female gamete, because female has only one type of a gamete. Whether it will be forming a female child, whether it will be forming a male child, female can only contribute the X chromosome, while in the female child the male is contributing the X chromosome, while when the male child, the particular male is contributing its Y chromosome. So the Y chromosome is determining the sex. That is why we can say the father will be going to determine the sex, not the mother. After this, the cleavage divisions. The process through which a single cell zygote undergoes the repeated mitotic divisions and changes into a complex multicellular structure without increasing its size is known as a cleavage division. Means rapid multiplications are going on, the number of cells are increasing, but the size of particular structure is not increasing. So zygote undergoes the division, and after many division it will become multicellular structure, but the size of that particular structure, the complete structure of the zygote, will not going to increase, and such kind of a division are known as a cleavage divisions. Now the blastomeres, the cells produced by the cleavage is known as a blastomeres. So normally the cells produced are known as a daughter cells, but here the cells produced by the cleavage division has a different name, that is known as a blastomeres. So after cleavage division the number of cells increases, and each and every cell is known as a blastomere. Cleavage starts as the zygote moves through the isthmus of oviduct towards the uterus, resulting in the formation of the 2, 4, 8 or 16 blastomeres. So this can be, again, the embryo with the 8 to 16 blastomeres is called as a morula. As the morula continues to divide, a cavity appears between the cells, and that is known as a blastocele. So when the morula again divide to form the many cells, the particular cavity appears between the cells of a morula, cells of the blastomeres, and this cavity is known as a blastocele. So after the formation of a blastocele, after the formation of this cavity, the morula is transformed into the blastocyst. So the name of the morula is changed to the blastocyst after the formation of the blastocele cavity, and this blastocyst moves further and it reaches the uterus. Now the blastomeres in the blastocyst are arranged into outer layers and inner layers. So blastocyst is having a multicellular structure, so it is having many cells. So the cells of the blastocyst, each and every cell is known as a blastomere, or I can say the blastomeres of the blastocyst arrange themselves into the two layer, outer layer and inner layer. The outer layer of the blastocyst are known as a trophoblast, and the inner layer of the blastocyst will be called as a inner cell mass. I will be coming to it with the diagram. Now this is the ovum, this is after the sperm penetrating the ovum, and this is the fusion of the nucleus, and this will become the zygote. So this is all we have talked about, the fertilization, fusion of the two different, and formation of the diploid zygote. This diploid zygote is a unicellular structure. This is a single cell. This is a single cell structure. The single cell structure undergoes the repeated mitotic division. So they will form the two cell structure. For the first time this will form the two cell structure. For the second time this will form the 4 cell structure, and then again 8 cell structure. But we can see that the size of this particular structure, the size of the particular structure, whether you can see here, here or here or here, the size remain constant, but the number of cells increasing. So we can also conclude that the size of a particular structure remains same, the number of cells increasing. It means the size of cell is increasing or decreasing. So we can see this cell is bigger in size while these cells are smaller in size. So during the cleavage division, the size of the blastomeres also decreases, but the size of an embryo or the particular structure will remain the constant. After zygote, the number of cells, the particular number of cells when it divide, each and every structure is known as embryo. It is known as a 2 cell embryo, it will be known as a 4 cell embryo, it will be known as a 8 cell embryo. And these embryo, the size of embryo remain constant during the cleavage division when it moves through the isthmus. So cleavage division starts when it moves through the isthmus. So this is the isthmus part of the fallopian tube, and cleavage will continue during the traversing during this isthmus part. Now, after 8 cell stage, or we can say 8 to 16 cell stage, is known as a morula. This stage, 8 to 16 cell stage, is known as a morula. So it is having a different name. It is now known as a morula stage. So 8 to 16 cell blastomere is known as a morula, and it is having a mulberry-like appearance. So that is why it is known as a morula. After formation of the morula, the cells divide again and form the many cells. When a particular morula cells divide and forms the many cells, there forms a cavity in between these cells. This cavity is known as a blastocyst. This is known as a blastocele. So this is known as a blastocele. And after the formation of this blastocele, this particular morula will be known as a blastocyst. So after the formation of the blastocele, the morula transform into the blastocyst. This is another structure. This blastocyst has reached the uterus. It has reached the uterus, and now this blastocyst will getting nutrition by attaching itself to the wall of a uterus. One more thing we can see in this blastocyst is that this blastocyst is having two layer. This particular blastocyst have two type of a cell. One are arranged inside and one are arranged outside. This is the morula transforming after the formation of this cavity, which is known as a blastocele cavity or blastocyst cavity. This cavity, after the formation of this cavity, this morula is transformed into the blastocyst. Now this blastocyst is having a two type of a cell. One is arranged in the outer layer and one is arranged in the inner layer. So these cells which are arranged in the outer layer are known as trophoblasts. The cells which are arranged in the outer layer are known as a trophoblast, while these cells which are arranged inside are known as a inner cell mass. So two type of a cell we can find out: the outer layer of a cell and the inner layer of a cell. Outer layer of a cell is known as the trophoblast, while inner layer of a cells which are attached to the outer layer of a cells is known as a inner cell mass. Now in further developments, these trophoblasts will go and attach to the wall of a uterus. So they will help in the process of implantation, and later on, after the implantation is complete, they will develop into the placenta. So trophoblasts will help in the formation of the placenta in the future. And the inner cell mass, these inner cell mass, all the cells are stem cells, and stem cells are those cells which are having ability to convert into the any type of a tissue or cell. So these cells will be going to form an embryo. So the trophoblast will be forming in placenta, and the inner cell mass will be forming an embryo in the future. So these are also known as an embryoblast. A proper embryo is formed by the inner cell mass. So this is also known as an embryo proper, which was formed by the inner cell mass. So inner cell mass will be developing into the proper embryo. Now implantation. So the trophoblast layer then gets attached to the endometrium, and the inner cell mass will get differentiated as an embryo. It means the trophoblast layer of a blastocyst get attached to the uterine wall or to the endometrium for the purpose of nutrition and growth and development. So this trophoblastic layer will help in the implantation. After attachment, the uterine cell divide rapidly and covers the blastocyst. As a result, the blastocyst become embedded in the endometrium of the uterus, and this is called as implantation, and it leads to the pregnancy. So after getting implantation, we can say that the pregnancy has started. So it takes place around 7 to 8 days after the fertilization. So after some days, after 7 to 8 days of the fertilization, the particular zygote moves to the isthmus and then to the uterine cavity. So it takes around 7 to 8 days, and after that the implantation is started. So implantation takes place around the 7 to 8 days after the fertilization. After implantation, the trophoblast starts secreting a particular hormone that is known as the HCG hormone. That is why we can detect the HCG hormone as a pregnancy detecting hormone. The female who is pregnant will be releasing the HCG. So that is why the after implantation the trophoblast starts secreting HCG hormone. If the the amount of HCG hormone is more in the particular the urine of a female, we can say that if we can find the HCG in the urine of a female, we can say the female is a pregnant. Now, which maintains the corpus luteum for the 9 months. I have already told you that the pregnancy will maintain the corpus luteum for the 9 months. But what actually maintains the corpus luteum during the pregnancy is the HCG secreted by the trophoblastic cells. This HCG will maintain the corpus luteum, and the corpus luteum will maintain for the 9 months. Now, this is how we can say this particular structure is a blastocyst. This blastocyst will go and attach, it will closer to the wall of the uterus. So this is how we can see this blastocyst structure. The outer layer are the trophoblast, inner cell layer are the inner mast cell. This is the endometrium and this is the myometrium of the uterus. This blastocyst going closer to the uterus, and this is how we can see this. And now after getting closer to this, the trophoblastic cells get attached to the wall of a uterus, and this is what we can say the attachment of the trophoblast to the wall of a uterus is known as an implantation. And implantation occurs at the 7th to the 8th day. So on the 7th to the 8th day, the implantation starts after the fertilization. So once it get attached to the wall of endometrium, this endometrium wall, this particular endometrium wall also proliferate, and it will form a layer, and ultimately this endometrium will proliferate and it will cover the complete blastocyst inside it. So that is why after the completion the implantation will get complete. So the endometrium wall also proliferate and covers the entire structure inside it. This is known as the completion of the particular implantation. One more thing we can see, this blastocyst structure also forms some finger-like projection. This is form some finger-like projection to get hold into this endometrium. So to get into this endometrium, this finger-like projection arises from the blastocyst or the trophoblastic layer. So this outermost layer, trophoblast layer, will form some finger-like projection. These finger-like projections will hold this particular structure inside the endometrium, and these finger-like structures are known as a chorionic villi. These are known as the chorionic villi. Now, placenta formation. After implantation, some finger-like projections appears on the trophoblast called as chorionic villi, which are surrounded by the uterine tissue and maternal blood. So this is how we can see. These are the blood vessels of a maternal tissue, and this is all is the endometrium. So this is myometrium and this is endometrium, uterine tissue. So when these finger-like projections get inside this uterine tissue, this is surrounded by the blood vessels. So that is why the chorionic villi, which is surrounded by the uterine tissue and the maternal blood. The chorionic villi and the uterine tissue become interdigitate with each other and jointly form a structural and functional unit between the developing fetus, developing embryo, or we can call it a fetus, and maternal body, and this structural and functional unit is known as a placenta. So the placenta is connected to the embryo through an structure that is known as an umbilical cord, which helps in the transport of the substances to and from the embryo. Now we will come to it. This is the structure which you can see. This particular part is this is the embryo, the developing embryo. This developing embryo, and this particular part where this chorionic villi, or we can say the placental villi or chorionic villi of the fetus, are interdigitated into the endometrial or the maternal tissue. This yellow colored part is the endometrium. So this is how we can see this part is the endometrium and this part is a chorionic villi. These both come closer, and this is how the interdigitations of this part takes place. So after the interdigitation of this part, the complete part from here to here, this complete particular structural unit is known as which is made by the maternal tissue as well as the fetal tissue. This particular unit made by these two tissue is known as a placenta. So this complete area in which the maternal tissue as well as fetal tissue interdigitate, this particular part is known as a placenta. So this is the placental structure. Now this embryo is connected to the placenta by a cord like structure, by this structure. So this cord-like structure is known as a umbilical cord, which carry the nutrition from this particular placenta to the embryo, and which carry the waste product of this embryo to the maternal blood. That is known as this particular structure is known as the umbilical cord. So to and fro, both the direction. So the nutrition can come to the fetus and the waste product can go from the fetus through this umbilical cord. Here also we can see that this is particularly a maternal tissue. This is the endometrium. This is uterine wall. This is endometrium, and this endometrium is interdigitated by this chorionic villi. So this villi and this particular maternal tissue will form an interdigitation. This interdigitation will form a complete structural unit that is known as a placenta. And through this placenta, there occur a cord-like structure which is known as umbilical cord, and this umbilical cord will carry the nutrition from the maternal blood to the fetus and can also carry the excretory part of this fetus to the maternal tissues. Now, function of the placenta. The function of placenta is the nutrition, respiration, excretion, and also sometimes it provide a passage for the antibodies like IgG antibodies to provide the defense mechanism to the developing embryo. So the placenta facilitates the supply of the nutrient to the developing embryo. Respiration, the placenta facilitates the supply of oxygen to the embryo and also removes the carbon dioxide from the embryo. Excretion, it helps in the removal of the waste material produced by the embryo and providing passage for the immunoglobulins like IgG. Now, formation of a hormone. This is another function of a placenta. Placenta also act as a temporary endocrine tissue. Similarly, like the corpus luteum, this particular structure, placenta, also can act as a temporary endocrine tissue. Temporary endocrine tissue means it will be going to release some of the hormones, and several hormones was released by the placenta like HCG. It is known as a human chorionic gonadotropin. Next is HPL, that is human placental lactogen. It can also release relaxin. It is releasing estrogen and progesterone and other hormones, etc. So mainly HCG, HPL, relaxin, estrogen and progesterone are released by the placenta. So it can also release certain hormones. Note that let us remember that HCG, HPL and relaxin are produced in the woman only during the pregnancy. These three hormones, HCG, HPL and relaxin, are only seen in the body of a female only during the pregnancy, during the period of a pregnancy, because these are released mainly by the placenta, and placenta is formed during the pregnancy. So these three hormones are mainly released during the pregnancy. Estrogen and progesterone can also be formed by the corpus luteum. So these two hormones can be seen in the female body before the pregnancy also, but after pregnancy we see some additional three more hormones, that is the HCG, human placental lactogen and the relaxin, and these three hormones are only produced during the pregnancy. Now, in addition, during the pregnancy the level of other hormones are increased several-fold in the maternal blood. So instead of those hormones, some more hormones will increase during the pregnancy period. The estrogen already has increased, progesterone has increased, but some other hormones like the cortisol, prolactin and thyroxine, these three hormones and some other hormones do also increase during the pregnancy period. So the amount of these hormones is increased in the female body during the time of a pregnancy. So what other hormones will increase during the pregnancy in spite of accepting the placental hormones? So the cortisol, prolactin and the thyroxine, these three hormones also increases. Progesterone, estrogen increasing because of the placental secretion, but cortisol, prolactin and thyroxine hormones also increases in the female body. What do the hormone do? So these hormones are responsible for the fetal growth. They are also responsible for the metabolic changes in the mother's body, and they are also responsible for the maintenance of the pregnancy. So maintenance of the pregnancy, for the metabolic changes and for the fetal growth, some other hormones will also increase in the body of a female. Now, after the gestation or embryonic development, so the development of the embryo is known as a gestation. So this period of the development is almost about 9 months. So the average duration of the human pregnancy is about 9 months, and which is also known as a gestation period, because it is a period of growth and development. So it is also known as a gestation period. So the gestation period and the embryonic development. Immediately after the implantation, the inner cell mass or the embryo forming cells differentiate into an outer layer called as ectoderm and the inner layer called as endoderm. So which we can see from this diagram also. I have told you, in this diagram we have seen this is the process of implantation. This is the particular blastocyst which is going to get implanted. During the implantation, when it is getting buried inside the wall of a uterus, when it is getting embedded inside the wall of a uterus, there occur a particular differentiation of an inner cell mass in which the two layers are formed. So similarly, these particular structure after this gestation period, we can see in this we can see immediately after the implantation, the inner cell mass differentiate into an outer layer called as ectoderm and inner layer called as endoderm. Soon after some time, the mesoderm soon appears between the ectoderm and the endoderm. So in between these two layers, the mesoderm also appears after some time. These three layers give rise to the tissue in the adult. So these three layers will be going to form a complete adult, and it will give rise to the different organs and tissue of an adult. It need to be mentioned that here that the inner cell mass contains certain cells which are known as the stem cells, and stem cells I have already told you which have the ability or potency to give rise to the organs and tissue, the other cells and organs and the tissues. So the inner cell mass is having the stem cells which will first differentiated as the ectoderm, endoderm and mesoderm, and these three different layers will form the different tissues and organs of a human body. Now this is how we can see. This is the morula, this is blastula. There are two layers. This layer is known as the endoderm, this layer is known as the ectoderm, and in between these two layers there are another layer that will be known as a mesoderm. So mesoderm will soon appear in between the ectoderm and the endoderm. So after that, the human pregnancy lasts till the 9 months, and what are the development that takes place during this 9 months? So in a human being, after 1 month of pregnancy, the embryo heart is formed. So the first organ that is formed is the heart. So the first organ, that is heart, which was formed during the first month of the pregnancy. The first sign of a growing fetus may be noticed by listening to the heart sound carefully through the stethoscope. So normally doctors listen to the heart sounds during the first month of the pregnancy. By the end of second month of the pregnancy, the limbs and digits will be going to get developed. After the 12 weeks, or we can say after the third month. So the pregnancy is a 9-month duration. It is a 9-month duration. This can be divided into the first 3 months, next 3 months and the last 3 months. So this 3-month time period is known as a trimester. So that is why it is known as the first trimester, it will be known as the second trimester, and it will be known as the third trimester. So that is why the first 12 weeks are known as the first trimester. Most of the major organ systems are formed during the first trimester. For example, the limbs and external genital organs are the well developed. They start forming in the second month, but they will be well developed after the first trimester, means after the 3 months of the pregnancy. The first movement of a fetus and the appearance of the hair on the head are usually observed during the 5th month. So in the second trimester, during the 5th month, the hairs on the head and the first movement of the fetus can be seen. So by the end of the 24th week, that is the second trimester. So second trimester means the completion of the 6 months. So by the end of the 24 weeks, in the second trimester, the body is covered with the fine hairs, eyelids are getting separated and eyelashes are also formed. By the end of 9 months of pregnancy, the fetus is fully developed and is ready to get delivered. So the time after this, the complete development will be known as the particular, after the 9 months of the pregnancy, the fetus is completely developed and is ready to get delivered outside the body. Now, the process of delivering of a baby is known as a parturition. So after the gestation period, vigorous contractions of the uterus at the end of the pregnancy, means after completion of the 9 months, causes expulsion or delivery of the fetus. The process of delivery of fetus is known as a parturition, or we can say that the process of childbirth is also known as a parturition. Parturition is induced by what makes the parturition, means how does the parturition is influenced or induced. So the parturition is induced by the complex neuroendocrine mechanism in which the neural impulses and the endocrinal impulses works to get the baby outside the female body. So it is also known as it is induced by the complex neuroendocrine mechanism, means both the system, neural system and the endocrine system, will help to move the baby outside the body of a mother. Fetal ejection reflex. The signal for the parturition arises from the fully developed fetus as well as the placenta. Means two things are giving the signals to the mother. First is the fully developed fetus itself and the placenta. These two things will be developing the signals for the parturition, which induces the mild uterine contractions called as fetal ejection reflex. So these two structures will induce the uterus to contract, and this is known as a fetal ejection reflex. Now, this triggers the release of oxytocin because of the placenta and because of the fully developed fetus. It triggers the release of oxytocin from the maternal pituitary. Oxytocin acts on the uterine muscles and causes strong uterine contractions, which in turn stimulate the further secretions of the oxytocin. We will be coming to it. Oxytocin is a main parturition hormone, and due to which the contractions in the myometrium arises and the baby is expelled outside. The stimulatory reflex between the uterine contractions and the oxytocin secretion continues, resulting in the stronger and the stronger contraction. So I will be come to it. This leads to the expulsion of a baby out of the uterus through the birth canal, that is known as a parturition. This process of delivering or expulsion of a baby outside is known as a parturition. Now we will be going to understand these two particular lines, what it is showing. So we have a particular diagram after that. Soon after the infant is delivered, the placenta is also expelled outside the uterus. So placenta did not remain inside. It will also be expelled outside after the delivery of a baby. One more hormone is helpful during the process of the parturition, that is the relaxin hormone. Relaxin hormone is also secreted by the placenta and also secreted by the ovary in the late phase of the pregnancy. So at the time of parturition, this relaxin hormone is secreted. What does this relaxin hormone do? This relaxin hormone relaxes the pubic symphysis to make the more space available for the fetus to move out. So it relaxes the pubic symphysis part so that it will make more space available for the fetus to get ejected outside the female body. Now this is how the neural and endocrinal mechanism takes place. A fully developed fetus and a placenta. A fully developed fetus and a placenta. The fetal ejection reflex arises from the fully developed fetus and the placenta. So they will develop a neural reflexes. They will send signals to the mother's brain. They will send signals to the mother's brain. So they will stimulate the mother brain. So when they stimulate the mother brain stimulates the pituitary gland to secrete the oxytocin hormone. So the fully developed fetus and the placenta will stimulate the mother's brain for the secretion of the oxytocin hormone. This oxytocin hormone will then carried in the bloodstream and will reaches the uterus of the mother. After reaching the uterus, it stimulates the contraction inside the uterus and the contraction inside the myometrium of the uterus. So during the structural description of the uterus, I have told you uterus is made up of 3 layers: perimetrium, myometrium and endometrium. Endometrium undergoes cyclic changes and myometrium undergoes the strong contraction which expels the baby outside during the parturition. So this oxytocin will lead the myometrium contractions and will lead the baby outside the body. So the oxytocin stimulates the uterine contraction and pushes the baby towards the cervix. So a hormone is functioning here, so it is a endocrinal reflex. So this is a neuroendocrinal mechanism in which both the neural impulses and the endocrinal impulses are functional. So now oxytocin is stimulating the uterine contraction. So that is why the head of baby is pushes against the cervix. So head of baby is pushes against the cervix. This will lead to the stretching of the cervix, and due to that stretching, the stretch receptors of the cervix send the nerve impulses from the cervix to the brain of a mother. So now these reflexes, which when released by the stretching of the cervix, will also lead the mother brain to secrete more amount of oxytocin. So this will lead to the increased amount of secretion of the oxytocin. This increased oxytocin will be carried through the blood to the uterus. This increased oxytocin will cause increased contraction. This increased contraction will again push the baby outside through the cervix, so the cervix will dilate more and cervix will stretch more. So the more stretching of the cervix will lead to the more nerve impulses from the cervix transmitted to the brain, and this will lead to the more stimulation again to the mother pituitary, which will release the more release of the, which will stimulate to release the more amount of oxytocin. So this cycle will continue until the cervix is fully dilated, and the strong contraction after the several cycles will lead to the expulsion of a fetus. So this is how this cycle will continue, and the one time it is when the cervix is fully dilated, the baby is pushed outside the body. So this is all about the parturition. So this is all about the human reproduction chapter, and now we will come to the NCERT textbook questions. So the first variety of question is a fill in the blanks type question, and the first question in the fill in the blanks type is, that is, human reproduce like asexually or sexually. Human reproduce and then fill in blanks. So they are asking, they have given you the option that human will reproduce asexual mode, by asexual mode of reproduction or the sexual mode of reproduction. So in the first class I have explained you that human is a sexually reproducing organism, means it involves the fusion of a gamete. So the answer will be a sexually reproduction, means it reproduces through the sexual mode of reproduction. So human reproduces sexually. Now the second is, humans are oviparous, viviparous or ovoviviparous. So I have already told you that the human that they give birth, the mother of human give birth to a young one, means they directly give birth to a young one. So they does not lay any types of eggs. So when a mother is giving birth to a young one, these kind of organisms are known as a viviparous types of organism. So human are viviparous in nature. So these I have already told you in the characteristics of the human, that humans are sexually reproducing and humans are viviparous. Now third, fertilization in the human. So what is the fertilization in the, what type of a fertilization can be seen in the human? So when we come to it, the fertilization in the human takes place inside the body of a female. So when the fertilization is taking place inside the body, then this kind of fertilization is known as a internal fertilization. So the fertilization in human is a internal type of a fertilization. Now the next, the male and female gametes are. So I have already told you that gametes are the haploid cell. So they are produced by the meiotic divisions. So the first meiotic division is a reductional division, and during that first meiotic division there is a reduction in the chromosomal number. So the gametes are haploid. So there were the male and female gametes, both are haploid cells. These are haploid in nature. Now the next, zygote. Zygote is formed by the fusion of two gametes, fusion of two different gametes, one from the male and one from the female. So when both the haploid gamete fuses, they form a diploid zygote. So the zygote in the human is diploid or haploid. So it is formed by the fusion of two haploids. So it is a diploid zygote. So this is diploid zygote. Now next, the process of release of ovum from a mature follicle is called as. So when a mature follicle, under the effect of the LH surge, on the 14th day, the mature follicle get ruptured, and due to the rupture of it, the gamete comes out. So that rupturing of a follicle, that releasing of ovum from the follicle is known as a ovulation. So this is ovulation. Now we come to next. Ovulation is induced by a hormone called as, so I have already told that ovulation is induced by, it is induced by the luteinizing hormone or LH. So the LH surge is responsible for the ovulation. Next, the fusion of a male and female gamete is known as. So the human is a sexually reproducing organism in which involves the fusion of a male and female gamete. So when the male and female gamete fuses, this particular process of fusion of two gametes is known as a fertilization, in which the male sperm is phagocytosed by the female is phagocytosed by the female gamete. Now, so the fertilization takes place in, so where does the, what is the actual site of a fertilization? Where does the fertilization actually takes place? Inside the female body. So the fertilization takes place inside the ampulla of the oviduct or fallopian tube. So the fallopian tube has three parts: isthmus, ampulla and infundibulum. And in the ampullary part of the oviduct, the fertilization usually takes place. Now, the zygotes divides to form which is implanted in the uterus, means the zygotes divide to form a particular stage, and that particular stage is responsible for the implantation. That particular stage is implanted inside the uterus, and that particular stage, we already know that zygote when divides to form a multicellular structure, and that multicellular structure I have told you is a significant stage which takes part in the implantation. That significant stage which is multicellular is known as a blastocyst. So that blastocyst, when it wants nourishment, it get attached to the wall of a uterus, and that process of attachment of blastocyst to the wall of a uterus is known as a implantation. So this is the blastocyst structure formed by the division of a zygote. Now, coming to the next, the structure which provides vascular connection between the fetus and the uterus, means there are two types of tissue involved. A particular structure formed by the involvement of the two different tissues, the fetal tissue and as well as the uterus is a part of a mother body, female's body, so maternal tissue. A particular structure formed by the fetal tissue and the maternal tissue, that particular structure is known as a placenta. So the structure which provides vascular connection, because the placenta is dipped in the maternal blood vessels, maternal blood, so it is known as a vascular connection. The vascular connection between the fetus and the uterus of a mother is known as a placenta. So this is placenta. Now, coming to the next, draw a label diagram of a male reproductive system. Now we will be coming to the different type of questions, that is a diagrammatic based questions. So they are asking to draw a label diagram for male reproductive questions. So I have taken the NCERT based diagram. In this diagram we can correlate all the male reproductive organs, like here we can see that this is the testis, and from the testis the vasa, this is the particular network of a seminiferous tubules known as a retrotestis. These retrotestis will be forming an the vasa efferentia taking sperms from retrotestis to the epidermis. This particular structure is epidermis. From this epidermis, the tail part of epidermis continues into a ascending tubule that is known as a vas deferens. This vas deferens taking the sperms to the abdominal cavity and loops around the ureter of the urinary bladder. So these are the structure of the urinary bladder and the ureter in the abdominal cavity. Then when it loops around, it will form a dilated part that is known as an ampulla of a vas deferens. This particular part is a vas deferens. The terminal part of the vas deferens is known as an ampulla. So this ampulla of the vas deferens join its ducts with the duct of these particular glands which are present on the sides of urinary bladder. These glands which are present on the sides of the urinary bladder or the lateral side of a urinary bladder are known as a seminal vesicle. So when the duct of seminal vesicle fuses with the duct of ampulla, it will form another duct, and that is known as the ejaculatory duct, which we cannot be able to see in this diagram, but there is the ejaculatory duct, and this duct will open into the prostate gland, the urethra around inside the prostate gland. So this structure is a prostate gland. The cut section of the prostate gland has been shown here, and after the prostate some smaller glands are present, the bulb-like glands are present, which are known as a bulbourethral gland, and after that this urinary bladder continues through the urethra to the penis, and inside the penis this urethra is known as a penile urethra, and from this urethra the sperms are taken outside the body of a male. So this is the particular structure, the diagram which is showing the complete male reproductive system. Now draw a level diagram of a female reproductive system. So on the contrary of the male reproductive system, the next diagram is of female reproductive system. In the female reproductive system, this is the only diagram in which we can explain the uterus, fallopian tubes and the vagina, all the ducts associated with it, and the primary sexual organ or primary reproductive organ of a female, that is the ovary. So in this diagram we can see the primary organs. These are the ovaries which are hanged inside the abdominal cavity, which are attached to the wall of a uterus and also to the pelvic cavity wall. Now after the ovary, we can see that there is two parts. First, this is the fallopian tube part. This is the fallopian tube which is comprised of the isthmus, ampulla and infundibulum. This infundibulum is having a finger-like projection known as a fimbriae, which helps in the correction of a ova when the ova is released by the ovary. When the secondary oocyte is released by the ovary, this particular ova is collected by these finger-like projections known as a fimbriae, and then they will transfer the particular secondary oocyte to the ampullary region of a fallopian tube. Now, second is the uterus. This is the particular cavity, this particular structure, the inverted pear-shaped structure which we can see This is the inverted pear-shaped structure. This inverted pear-shaped structure is known as a uterus. It is having a cavity within it that is known as a uterine cavity. The uterus is made up of three. It is classified as a uterus is classified into the three particular parts. That is the fundus. The uppermost part is known as a fundus. This particular part from here to here known as the body of a uterus, and beneath it we can see the narrow canal. This narrow canal is known as the cervix of a uterus. So in this diagram, in this particular diagram, we can see all the particular structures, and after the cervix, beneath the cervix there is a long and narrow canal that is known as a vaginal canal or the vagina. So which will end up on the vulva or external genitalia of a female. So this in this particular diagram we can explain the complete female reproductive system. Now after that, write two major functions of each testis and ovary, means both are the primary reproductive organs, and we have to define the two major functions for both of them. So first of all, the function of a testis. So primary reproductive organ has two functions. First, to form a gamete, and next to form a particular, to secrete a particular hormone. That hormone will help in the particular the hormone will be helping that particular primary reproductive organs to for the process of a gametogenesis. So they produce a male gametes by the process called as spermatogenesis. So it is helping in the gametogenesis process, or they are helping in the formation of a sperm. So the first function is the formation of a gamete. Next, they are having a Leydig cells. The testis is containing a Leydig cell within it, and this Leydig cell, which is present inside the testis or near the seminiferous tubule, secrete the male sex hormone that is known as testosterone, and we can also broadly call as androgens. So that can be testosterone or androgen. Both the answers are correct. So the primary functions of the testis are the formation of a gamete as well as the secretion of a hormone which is responsible for the formation of a gamete. So these two things. Now coming to the ovary. Similarly, like the male reproductive, the primary reproductive organ of a male is testis, the primary reproductive organ of a female is the ovary. So the ovary is also having the same functions. The ovary, the first function, the primary function of ovary is the formation of the female gamete, that is ovum, by the process of a oogenesis. So the gametogenesis in the female is known as a oogenesis. The growing follicles, the growing Graafian follicles secrete a female sex hormone that is known as a estrogen, and an estrogen is responsible for the ovulation. Again, it will rise the amount of LH, the level of LH, and LH is responsible for the ovulation. So gamete producing hormone is a LH, but here the estrogen is also secreted along with that. So the growing follicle secretes the female sex hormone that is known as the estrogen. We can also use progesterone also. The progesterone is also released by the ovary itself. So function of ovary is formation of a gamete and secretion of a hormone which is responsible for the gamete formation. Now next, describe the structure of the seminiferous tubule. So inside the seminiferous tubule and outside the seminiferous tubule, we will talk about this structure. Seminiferous tubule is made up of a layer of a male germ cell and large Sertoli cells. Means the seminiferous tubule inside is lined by two types of a cell. First is the spermatogonia or the male germ cell, and the another one that is a bigger in size that is known as the Sertoli cells. Sertoli cells are also known as the nurse cells because they provide nourishment to the growing sperms or the growing spermatocytes. So male germ cells undergo spermatogenesis, means male germ cells are also known as the spermatogonia. Spermatogonia undergoes spermatogenesis to produce spermatocytes, spermatids, and ultimately convert them into the sperms. So these all the structures, whether it is spermatogonia, whether it is male germ cell, whether it is spermatocyte, whether it is spermatid, or whether it is sperm, these all structures get nutrition from a Sertoli cell. So Sertoli cells providing nutrition, that is why it is known as a nurse cell. Now, region outside the seminiferous tubule. This is all about the inside seminiferous tubule. Now, region outside the seminiferous tubule is called as interstitial space. Means the region between two different seminiferous tubule is known as the space between two seminiferous tubule is known as a interstitial space. And the interstitial space have connective tissues which includes the blood vessels and the Leydig cells. One more cell is there, that is an immunocompetent cell, which is not more significant, so it was not used here. So which is included the blood vessels as well as the Leydig cells. Now what is the importance of these Leydig cells? So the Leydig cell synthesizes and secretes the male sex hormone. These are known as a androgen, or which we can also known as a testosterone. The principal hormone that is used inside the androgen category is the testosterone. So both are the same. We can use androgen or we can either use testosterone also. So these are the this is about the Leydig cell. This is about the male germ cells and the Sertoli cells. So this is all about the structure of the seminiferous tubule. Now, this is the particular NCERT diagram in which we can see that this is the particular structure, the cross section of the seminiferous tubule, and inside the seminiferous tubule we can see this is the outermost covering of the seminiferous tubule. Inside that we can see the spermatogonias. These spermatogonium are also known as a male germ cell. These male germ cells, when divide, they will form a primary spermatocyte. These male germ cells, when divide, they form a primary spermatocyte. These primary spermatocyte, when the primary spermatocyte divide, they will form the secondary spermatocyte. And then the secondary spermatocyte will convert into the sperm, which get nutrition from these cells. These are known as a Sertoli cells. These cells in between the spermatogonia and the spermatocyte is known as a Sertoli cells. So these cells are providing nutrition to each and every cells along with them, that is the spermatogonium, primary spermatocyte and secondary spermatocyte. Now, when they transform into the spermatid also getting nutrition from the Sertoli cells. And when these spermatids are transformed into the spermatozoa, these spermatozoa or sperms are also getting nutrition from these Sertoli cells. This is the particular structure. This is the diagram of the